I'm going to walk you through a complete RNA-seq analysis pipeline implemented in Python. This pipeline automates the process from raw sequencing data all the way to biological interpretation through visualization. Let's start with the overall structure. The pipeline is organized into 6 main steps, and each step corresponds to a key stage in RNA-seq analysis. First, we set up logging and configuration. The script reads a config.yaml file, which defines all the important paths, such as where raw FASTQ files are stored, where results should be saved, and where the reference genome is located. This makes the pipeline flexible and easy to reuse on different datasets. Step 1 is quality control using FastQC and MultiQC. FastQC evaluates the quality of each sequencing file, checking things like base quality scores and adapter contamination. Then MultiQC aggregates all reports into a single summary, so we can quickly assess the overall data quality. Step 2 is read trimming using trim_galore. This step removes low-quality bases and sequencing adapters. Clean data is essential because poor quality reads can negatively affect downstream alignment and quantification. Step 3 is alignment using star. Here, the cleaned reads are mapped to a reference genome. The output is a sorted BAM file for each sample, along with gene level counts. Star is chosen because it's fast and accurate for RNA-seq data. Step 4 uses feature counts to quantify gene expression. It takes the aligned BAM files and counts how many reads map to each gene based on a gene annotation file. The result is a gene count matrix, which is the foundation for differential expression analysis. Step 5 performs differential expression analysis using edgeR. The Python script calls an external R script, passing in the count data and metadata. edgeR then identifies genes that are significantly up or down-regulated between conditions. Let's quickly walk through this edgeR script for differential expression analysis. First, the script reads 3 command line arguments: the counts file, the metadata file, and the output file. This makes the script flexible and easy to plug into a larger pipeline. Next, we load the edgeR library and import the data. The counts matrix contains gene expression counts, and the metadata provides sample information, especially the experimental condition. We then define the grouping variable based on the condition and create a dgelist object, which is edgeR's core data structure. After that, we filter out lowly expressed genes using filter by expr. This step improves statistical power by removing noise. Then we normalize the data with calc norm factors to account for differences in library size and composition bias. We also calculate logCPM values, which are exported for visualization steps like PCA and heatmaps. Next, we build a design matrix and estimate dispersion, which models biological variability. Then we fit a generalized linear model and perform a likelihood ratio test. Finally, we extract all differential expression results and save them to a CSV file. In summary, this script takes raw counts and produces statistically robust differential expression results ready for interpretation. Finally, step 6 focuses on visualization. This includes 3 key plots. First, a PCA plot, which shows how samples cluster based on their gene expression profiles. This helps us check whether biological replicates group together as expected. Second, a heat map of the top differentially expressed genes. This highlights expression patterns across samples and can reveal biological trends. Third, a volcano plot, which combines fold change and statistical significance to quickly identify important genes. Overall, this pipeline is modular, reproducible, and scalable. Each step is clearly separated, making it easy to debug, modify, or extend. For example, you could swap out the aligner, change the normalization method, or integrate additional downstream analyses. In summary, this code provides a full end-to-end RNA-seq workflow, from raw data to biological insight, using a combination of Python for orchestration and R for statistical analysis.